Yeah.
Prashant. Hello, Prashant.
हाँ सर.
Prashant, it's getting late. Make expert as co-host.
Yes sir. Yes sir.
Hello, Prashant.
Yes sir.
Prashant, it's getting late. Make expert as co-host.
Yes sir. Yes sir.
Hello, Prashant.
Yes sir.
Prashant, it's getting late. Make expert as co-host.
Yes sir. Yes sir.
Hello, Prashant.
Yes sir.
Night, sir.
Yes sir. Hello.
हाँ प्रशांत काही प्रॉब्लेम आहे का तर तुम्ही सर इंट्रोड्यूस करून द्या गेस्टला विल स्टार्ट देन
हो हो. आणि hello.
हाँ यस सर
Yeah. Good afternoon, everybody. The the expert for today's talk is Dr. Anil Dahuja. I welcome all the expert and the
सर दोन पैकी एक तुमचं कॉम्प्युटर किंवा मोबाईल थोडस म्यूट करा सो दॅट देर विल नॉट बी एनी इको. Okay, yes.
Yeah. So I welcome you all and very good afternoon. The webinar on advances in basic plant research in relation to climate change. Today's speaker is Dr. Anil Dahuja, who is Principal Scientist and Professor, Division of Biochemistry, Indian Agricultural Research Institute, New Delhi. He'll be talking on the topic of enhancing the bioavailability of phytonutrients, a step forward towards achieving sustainable nutritional security under changing climate. Dr. Dahuja joined the Division of Biochemistry in 97 and presently he is holding the post of Professor, Division of Biochemistry, and is a Principal Scientist. Dr. Dahuja's field of specialization is bioactive compound and bioprocessing for plant and human health. He has got a list of different in-house projects in which he is working in the capacity of either principal investigator or co-principal investigator. He has more than 80 publications to his credit in the different in the NAS-rated journals, and the in-house projects are particularly on the genetic modification of soybean for improved nutritional and flavor quality, development of nutraceuticals and functional foods from horticultural produce and cereals and pulse-based convenient food products, development of improved technologies for higher yield and quality in selected food crops, seed quality enhancement by processing, packaging, and storage options in high volume seed crops, and development of varietal and hybrid technologies of pearl millet for higher yield and nutritional improvement. Dr. Dahuja has developed construct for overexpression of isoflavone synthase and downregulation of flavone 3 hydroxylase for the metabolic engineering of phenylpropanide pathway in soybean for developing high isoflavone soybean. He has also developed a cost-effective and eco-friendly microwave and enzyme-assisted extraction method and optimized the extraction for anthocyanin without loss of stability and functionality from black soybean seed and black carrot. Dr. Dahuja has been awarded as the Best IRI Teacher Award in the year 2012, Young Scientist Award in the year 2013. Dr. Dahuja also presently has got 3 important projects funded by DBT and National Fund for Basic and Strategic Research of ICR. Is working on the RNAi silencing and production of low phytate soybean, and so it is our fortune that he is available today for this webinar. I welcome Dr. Dahuja and request Dr. Dahuja to proceed with his presentation. Dr. Dahuja, please.
Thank you, Dr. Nayak. for the elaborate introduction. Am I audible?
Yes, yes.
Yeah. Okay.
Yeah,
Thank you,
yeah.
Dr. Nayak, and all the organizers. I can see Dr. Pawan Kulwal, the joint organizing secretary, and then Dr. Lokhande is there, the coordinator. So I thank you all for inviting me for this particular lecture. So although personally I would have loved to give this lecture in your campus, so I had been to your campus before also, and it was really a wonderful experience giving lecture in that hall which was full of students. So it is altogether different feeling. But nonetheless, the circumstances which we are in, so we don't have the other option. So I will be delivering my lecture through online mode in this webinar. And I must congratulate the organizers for selecting this topic for the webinar, the advances in the basic plant research in relation to the climate change, because this is a very timely webinar which has been organized, because this is the need of the hour, because climate change, it is again, earlier we were thinking that it is going to enhance the yield of this of certain crops, especially the C3 crops, but now the research, it is showing that the climate change, it may have the adverse impact on the nutritional quality of the various food crops, especially the C3 plants. So in that way, this is very, very important, because nutrition, you in the times of the COVID, you can see that it is really a need of the hour to have good nutrition, because it enables us to fight with various kinds of diseases. So with this brief introduction, I would like to have your permission to share my slides so that I can proceed further. My slides are visible to all?
Yes, sir. Yes, sir.
So, as has been told by Dr. Nayak before, the topic which has been given to me for this particular lecture, it is enhancing the bioavailability of the phytonutrients, which is a key step forward towards achieving sustainable nutrition security under the changing climate. So, I once again and thank the organizer for giving me this opportunity. And as you know that now we are in the COVID-19 pandemic situation, and I will just give you a highlight what kind of nutrition which is being recommended during these trying times. So as you can see that people with strong immunity levels are less prone to the infection, and that's why the general dietary recommendation which are coming from experts is that diet should be rich in the immunity boosters, which the like say minerals like zinc, vitamin, especially the vitamin D, it is having a strong correlation as far as the incidences of the infection is concerned. And then there are a lot of recommendations about the phytonutrient rich herbs, and then of course the proteins, because you know that to fight any kind of infection we need the antibodies in the form of proteins. So the enhancement of the protein, enhanced intake of rather proteins, it is going to be beneficial to fight these infections. But this is the infection and the situation which is now, but again in the future you might be encountering different kinds of infections. We don't know what kind of shape this is going to take. So that's why the nutrition, it becomes of prime importance. So let's see where we are now at present as far as the protein and the micronutrient malnutrition it is concerned. Because I have in the previous slide I have told you that the protein and the protein requirement and the requirement of the micronutrients, it is of utmost importance to fight any kind of infection. So let's see where we actually are at present. So as you can see the data which is presented here, the hidden hunger affects around 2 billion people, or every one in three persons it is suffering from the hidden hunger. And in India, the reports say that over 80% of the adolescents suffers from the hidden hunger. And when it comes to the number of undernourished people globally, we can say that there are more than 600 million people. In India, it is 189.2 million people, approximately 14% of the population. And then in India, if you see the stunted, underweight, and the wasted children, so you can see the percentage, it is very, very high. And looking at the importance of the protein and the micronutrient malnutrition, the sustainable goals they have been proposed. And here I would like to pinpoint the about the second sustainable development goal, that is the zero hunger. And if we really want to achieve this goal, so we need to make necessary changes now, because otherwise it won't be easier for us to achieve these goals. So let's see, in achieving this a very important goal, that is SDG 2, what are the major foreseeable stumbling blocks in our way? So the first and the foremost, it is the growing human population. As you can see that by 2050, so population, it is going to be more than 9 billion. And then the resources which are available to us, whether it is in the form of land or water resources or the nutrients, so they are depleting day by day. And poverty, it is again another factor which is going to be very, very crucial, rather a very crucial hindrance, which in our way to attain the nutritional security. And the last one but not the least, it is the climate change, because I have specifically included this because it aptly covers the topic of the webinar, that is the advances in the basic plant research in relation to the climate change. So let's see how this climate change, it is going to affect the nutrition of the various crops. So climate change, I will take it as a multifactorial stress. Just like any other biotic and abiotic stress, climate change, it is also a kind of stress. And the various studies they have been performed in relation to the climate change and different conditions in the controlled environment chambers, in the open top chambers, and then free air carbon dioxide enrichment experiments. I won't go into the details of these kinds of experiment, because I believe that in you had an excellent lecture in the morning, and the speaker had eloquently given a view of the climate change and how it is going to change the productivity and the quality of the grains. So I won't go into the details of this, but still, to maintain the continuity, I would like to highlight this thing, and that because of the increasing concentration of the carbon dioxide, the elevated concentration of the carbon dioxide, it has been shown to enhance the plant growth, and that's why the biomass production and the yield of the crops, especially the C3 plants. Again, I am emphasizing that this is mainly pertaining to the C3 plant, because C4 plants, they have their own mechanism to concentrate the carbon dioxide, so there's not much of effect on the of the enhanced carbon dioxide concentration on the C4 plants. And because the plant growth and the yield, it is increasing, so that's why there is increased demand for the nutrients and the water, because this is not going to be possible without the nutrients and water. So that's why you can see that more and the more of nutrients, they are being taken up from the soil, and that in turn lead to this lower availability of the nutrients in the soil. So soil health, it is also going to change as far as the climate change is concerned, as far as the elevated carbon dioxide concentration is concerned. So not only with respect to the nutrients, but the number and the diversity of the microorganism, it is also going to be severely affected by the elevated carbon dioxide concentration, and that in turn will affect the availability of the nutrients. And because more and more nutrients, they are being utilized for the growth and development of the plant, so the accumulation of the nutrients in the grain, it is going to reduce, as was emphasized in the forenoon's lectures as well. And because of all this, so what are the nutritional consequences? So we have seen that the because the night whenever you have the elevated carbon dioxide concentration, so there will be more synthesis of the carbon based non-structural carbohydrates. So C to N ratio, it will be increased. And because a cell, it has to maintain the C to N ratio, so increase in the carbon, it has to be compensated by the decrease in the nitrogen level. So nitrogen level, they have been found to decrease at the elevated carbon dioxide concentration. Otherwise also, in earlier also we had reports in the literature where they have shown that so if you tinker with the nitrogen metabolism pathway, the carbon metabolism pathway, it gets affected, and in reverse also it is true. So there will be be decrease in the nitrogen levels, and that in turn will lead to the decrease in the protein levels, and of course the reduction in the levels of the micronutrients like iron and zinc. It is going to happen, and the reason which has been given for this particular decrease in the micronutrients, it is that the carbohydrate dilution. So as I mentioned, that there will be more and more synthesis of the non-structural carbohydrates. So concentration of the carbohydrate, it is increasing, so that in turn is leading to the dilution of the micronutrients. But again, this theory, it has been questioned, because if this was true, so then the reduction in the level of most of the micronutrients, they should have taken place in the same proportion, but it has not been observed. So the reduction or the decrease, it is much more in case of iron and zinc as compared to the other micronutrients. So this clearly refutes this theory, that is the carbohydrate hydrate dilution theory. And this is the possible mechanism which has been given for this particular effect which we have seen, that elevated carbon dioxide, it is leading to the lower concentration of the nutrients. So as I have told you that enhanced CN2, when elevated carbon dioxide is there, so there will be enhancement in the C/N ratio, and because of that there will be reduced stomatal conductance also. The elevated carbon dioxide, when it will also lead to the reduced stomatal conductance, because you all know that there is a trade-off between the exchange of the carbon dioxide and the water vapors. The carbon dioxide, it goes inside for photosynthesis, while the water vapors, they come out for transpiration. And it has been found that under elevated carbon dioxide concentration, the stomatal conductance, it is going to reduce. This is because the functioning of the guard cells, it gets affected because of the reduction in the accumulation of the ions and the metabolites. Because as I told you, the elevated carbon dioxide, it is going to affect the metabolism in very drastic way, so that will in turn lead to the changes in the ions and the metabolites, and those changes in the ions and the metabolites, it will affect the functioning of the guard cells. And because of that the stomatal conductance and stomatal aperture, and even sometimes it has been found that the stomatal development and the stomatal density, it is also getting changed under the elevated carbon dioxide conditions. And because of this, the transpiration rate, it is getting reduced. But one of the negative impact of this is, of course, the reduced transpiration, it also helps in the enhanced drought tolerance to the plants, and enhanced water use efficiency to the plant. But in relation to the nutrients, so whenever the transpiration rate, it gets reduced, so it will lead to the reduced flow of the nutrients, and that could be one of the reasons for the lower accumulation of the nutrients. So as I told you that many theories, they have been put forward, and many types of experiments under different locations, they have been conducted, but they got the contrasting results. So when they worked on the phase or the open top chambers or the controlled environment conditions, there were a lot of variations and diversity between the results. And you know, now in case of drugs, a meta-analysis approach, it is being done, because you take any research, there will be thousands of the paper which will be supporting that research, and there will be hundreds and thousands of the paper which will be refuting those claims. So now what is the truth? And for to reach to the truth, this meta-analysis, it is being done, and same it has been done in case of to study the effect of the elevated carbon dioxide concentration on the human nutrition. And one group based in the Japan, Australia, and the United States, they carried out this study, and this work, it was published in the Nature in 2014. And what they did was, because why there was variation in the results, was one of the prime reason was the sample size, it was very less. And through meta analysis, they wanted to have a big size of the data. And that's why what they did was, they took 6 different crops: rice, wheat, maize, soybean, field pea, and sorghum. Sorry. And then they did the experiment at 7 different locations, and Japan, Australia, and United States. Of course, the countries, there were 3, but locations, there were 7. Within the countries, different sites, they were selected. And then they could compare the 143 they could did the 143 comparison at ambient and the elevated carbon dioxide concentration, and this was the concentration which was chosen by the group, that is between 546 to 586 ppm. And you all must be knowing that it is predicted that by 2050 the concentration of the carbon dioxide in the atmosphere, it is going to be 550 ppm. And that's why, and of course till at the end of the century, it has been predicted it is going to be more than 900 ppm. So that's why they have chosen this particular concentration of the elevated carbon dioxide, and they carried out this study. And what they observed was that yes, there is a decrease in the accumulation of the nutrients under the elevated carbon dioxide concentration, and all those claims which were refuting this theory, they have been now put aside, and now it is believed that the elevated concentration of the carbon dioxide, it is going to decrease the accumulation of the nutrients through this experiment, through this meta-analysis approach. Now we are now very much sure that the elevated carbon dioxide concentration, it is going to decrease the level of the nutrients. So what are the strategies which we have which can enable us to rather which can prepare us to face this particular challenge? And we have many approaches which I have listed here. And the first one is the role of metabolic engineering. So we can go for the development of the biofortification, biofortified crops using the genetic manipulation. And then the breeders, they can develop certain varieties in which nutrient levels are less sensitive to the elevated carbon dioxide. So in the experiment which was done by that group which I presented in the last slide, so what they have observed is that is within the varieties also there is a difference. So in some cases there is less reduction in the level of the nutrient, in some varieties there is more. So that's why the breeders, they should look for varieties which lead to the lesser reduction in the levels of the nutrients in response to the elevated carbon dioxide concentration. And of course the agronomic fortification, it is another strategy which is there. And the next one is the supplementations. You can supply the nutrients you can go for the blending of the flours and the oils and all those things. And then the plant growth promoting bacteria because as I said that the soil health, it is also going to reduce under elevated carbon dioxide concentration. So if we can have certain plant growth promoting bacteria in the soil, so that will enable the enhanced uptake of the nutrients from the soil. So these are the 5 strategies, but these strategies, they are time consuming ones. So but currently what we can do is in say next 2-3 years is enhancing the bioavailability of the nutrients, because there is no point in having nutrient dense crops unless until these nutrients, they are bioavailable in the human body. So we, by using n number of approaches, you can go for the development of the nutrient dense crops, but if they are not bioavailable, so then there's not going to be any improvement in the human health. So that's why I consider the bioavailability of the nutrients of utmost importance as far as enhancing the human health it is concerned. So let's see how we can what this bioavailability of the nutrients is, and what strategies we can apply to enhance the bioavailability of the nutrients. So if you see the human body, I consider it as a machine. So just like a machine, it requires the fuel for its functioning. Similarly, human body, it requires fuel in the form of diet. And the genome, it is the engine. So just like the quality of the machine, it is determined by the quality of the engine. So same way, our quality, it is determined by our genome. So whether our genome has the sufficient information, the sufficient competency and the capability to convert the diet into the useful metabolites which can affect our health in the positive way. So if genetic information stored in our genome, if it is having all these features, so naturally we are going to be healthy. but there are certain external factors also which are playing an equally important role. So in the subsequent slides we'll be discussing all these things. So if you just see what in our diet, what kind of nutrients they are present. So basically we can divide them, the dietary components, into nutrients and the non-nutrients. The nutrients, they can be further divided into macro and micronutrients. The macronutrients, you all know, it is the protein, carbohydrate, lipids, and the nucleic acid. But for macronutrients, the bioavailability, it is not a major issue, because during the course of evolution, the human beings, they have evolved certain kinds of receptors and the transporters which ensure the very high bioavailability of the macronutrients. But for micronutrients like vitamins and minerals, the bioavailability is still an issue. And both these macro and micronutrients, they are playing different kinds of nutritional roles that are required for sustenance of the human beings. And when it comes to the non-nutrients, or we call them as the phytochemicals or the phytonutrients, for example, the anthocyanin, carotenoids, tocopherols, and flavonoids. So you all know they belong to the secondary metabolite category, and secondary metabolites, they are not required for the primary growth and development of the organism, but they are required for the quicker adaptation to the unforeseen situations. And they are providing the benefits which are beyond the nutrition, and they are required not for sustenance but for the maintenance, and that's why they are also called as the lifespan essentials. So let's see how our diet, it has evolved over during the course of evolution. So initially, and the concept was to have the adequate nutrition. This was a classical concept, where the people, they wanted to have more of the carbohydrate, proteins, vitamins and minerals, in order to ensure the what we call a balanced diet. But now the people, they are becoming more and more aware about the relationship between the diet and the health, and the various kinds of bioactive components which are present in the diet that can have a positive influence on our health, or the negative influence is also possible. So people now, they are moving from adequate nutrition to the optimal nutrition. So this is the modern approach, and where it has been seen that the bioactive components present in the diets, they should be exploited, they should be used for the development of the functional foods, and those functional foods then can have various kinds of health promoting and disease preventing activities. So this concept of the functional food, it is not new. It is very old concept. Almost 2,500 years ago, the Hippocrates said that let food be the medicine and medicine be the food. And then again, in year 1993, the Japan, there was a very big news at that time. The Japan explores the boundary between the food and medicine. And what they did was they developed a hypoallergenic rice, or also known as the fine rice, which was approved for the first food for specific health use, the foshu, the commonly it is known as. And then at that point, the path of the nutrition science came to the full circle. So it all started with the Hippocrates, and then it ended in the development of the first functional food, that is the foshu. And this is how we define the functional food. So it should have an positive effect, uh, and it should be either used for maintenance or improvement of the state of health, or well-being, or reduction of the risk of the disease. So these are the different categories of the functional food. It could be a basic food, carrots, odd bran cereals, and even the soya bean. It is nowadays the crop which I am working on. It is also considered as a functional food because it contains many bioactive molecules. And then certain processed food which are having added ingredients. So we now have the oil which are rich in the omega-3 fatty acids. We have certain milk which has been uh, fortified with certain bioactive components. The juices, they are fortified with various kinds of components. Just I have given this example, the calcium rich fruit juices. So all these processed food, they are also considered as the functional food. And then many times the bioactive because now we have a lot of information available with us, what kind of bioactive components they are actually responsible for the the health beneficial or disease preventing effect. So we can go for the isolation purification of those bioactive components and then use them in certain excipient, and we can use prepare certain formulations, and such kind of components, they have been given a definite the specific name, that is the nutraceutical. So you all must be knowing about that. So these are some of the bioactive components which have been reported in various foods, starting from the omega-3 fatty acids, and then isoflavonoids in soybean, and then many phenolics and even some alkaloids. And here I would also like to mention about the prebiotics, which are present in the whole grains. So a lot of emphasis it is being given for the consumption of the whole grains because of the their richness in the various types of prebiotics. And these are the mechanisms by which the functional foods or the bioactive components, they are preventing diseases. I won't go into the details of all these because these are the common things you all must be knowing about that, because most of the functional food components, they have the antioxidant activity, and they change the balance of the hormones, and then inhibition of certain enzymes. So these are the different ways by which the phytochemicals or the bioactive components, they can influence. But the problem with the current nutraceutical research is that it is very very difficult to extract the bioactive component, because they are secondary metabolites, they are always present in the very less amounts in the foods. So their extraction procedure is going to be very very difficult, and always you will find the yield obtained, it is very less. And also during the extraction process, sometimes you may have the change in the functionality of that bioactive component, because stability, it is very very less. So it is always going to be a very big trade-off as far as the yield and the stability or the functionality it is concerned. And then you don't have the delivery vehicles to transfer these bioactive components from the diet into the our system. And then there's lack of convincing scientific proof of functionality. So that is also because it is not easier to make people believe about the health claims which are there. Of course, in the COVID-19 situation, the people are believing more and more, even if they are miss, but they still believe it. Sorry. And then the another deficiency in this field, it is the reduced bioavailability of the bioactive component. So this is which we'll be discussing in more detail in the subsequent slides. So if you see what this bioavailability is, the bioavailability, if I have to define it in two lines, I would say that the rate and extent to which a bioactive compound is absorbed and becomes available at the site of action. So this is very very important. So it should be available at its physiological site of action, because only then it will be able to provide the desired benefit to the consumer. And then or we can define it as the fraction of the ingested nutrients that reaches the systemic circulation and is utilized by the body. Because ultimately, if you want to study the bioavailability, what you will do is you will study it, the level of the bioactive component in the systemic circulation. And this bioavailability, it encompasses 5 steps. It is a not a simple process, it is very integrated process involving liberation, absorption, distribution, metabolism, and the excretion. The commonly we call it as the LADME. So what these steps are, we'll see in a few minutes in the subsequent slides. So how do you measure this bioavailability? So as you know that nutraceuticals, they are very similar to drugs. For drugs, we have a well-defined mechanism to study the bioavailability. So on the left-hand side on in the slide, you can see the type 1, type 2, type 3, and type 4 drugs. So it what actually these types means are the type 1 drugs are having the high solubility and high permeability. So that means their liberation, if it is there, so then the solubility in the gastrointestinal tract, it will be more, and they will be permeable in the enterocytes. so they are going to be more bioavailable. So for such type of drugs, there is no issue as regard to the bioavailability it is concerned. And coming to the second type, that is the low solubility and high permeability. So these types of drugs, they are not soluble in our gastrointestinal tract, but they have the ability to pass through the enterocytes, the cells present in the intestinal lumen. So that's why this type of drugs, they are usually the hydrophobic drugs, because if drugs, they are hydrophobic, so naturally their solubility in the gastrointestinal tract, it is going to be less. The type 3 type of drugs, they are actually high solubility and have the low permeability, so they belong to the a hydrophilic type of drugs, because hydrophilic drugs, they will be soluble in our gastrointestinal tract, but because of their hydrophilicity, so they won't be able to pass through the intestinal mucosa, and that's why the permeability it is low. And the next category, it is the type 4, which is low solubility, low permeability. So such kind of drugs, they are never going to be bioavailable, because they are not soluble in the gastrointestinal tract, and they don't have the ability to pass through the lumen. So this is the nomenclature which has been, classification system, or rather I would say, which has been proposed, and which is well established in case of drugs. And now recently, for nutraceutical also, the researchers, they have come up with this kind of classification, which is known as the BAT classification. BAT stands for: B is for bioaccessibility, A is for absorption, and T is for the transformation. And for a bioactive component or the nutraceutical to be bioaccessible, it should first be released from its food matrix, because you know that these bioactive components, they are embedded in the food matrix. Take any example, the carotenoids or phenolics, they are always bound in the complex network of dietary fibers, and sometimes the cell walls. So it is really difficult to liberate these bioactive component from the food matrix, and once it is liberated, it should be soluble also. Unless until it is soluble, so it is not going to be bioaccessible. And once the solubility is there, so then, because in the gastrointestinal tract there will be n number of metabolites present, so they may interact with your bioactive component, so this may also influence the bioaccessibility. So liberation, solubility, and interactions, they determine the bioaccessibility. And once the bioactive component it is bioaccessible, so then it has to be absorbed, and then and finally it will be transformed either through metabolism or the chemical degradation. So I'll discuss in detail each of these three aspects, but for the time being, you just remember how actually we represent this bioavailability in case of nutraceutical. So here you can see the B star, it actually represents the bioaccessibility, A for absorption, T for transformation. The negative signs here, it's showing that it is limiting. The positive sign shows that it is non-limiting, and the as it shows that which factor it is actually making the bioaccessibility non-limiting, rather limiting, because it is negative, so it is limiting. So as here stand for the solubilization. So this example I have given you for a lipophilic nutraceutical, like say curcumin. Curcumin, you all know it is a lipophilic molecule, and because it is hydrophobic, so it is solubility is poor, so that's why it is limiting. And absorption, it is not going to be a problem, because it is going to be easily permeable through the membranes, so that's why absorption it is non-limiting. And transformation, it is again a negative, that means it is again limiting, because this curcumin it is susceptible to metabolism. So M here stand for the metabolism, and it is also very prone to degradation by various chemical reactions. So this is how we represent the bioavailability of the curcumin. And then on the left hand side of this slide, so you have one more graph which actually shows you that how you actually determine the bioavailability. So in this graph there are different terms. You have Cmax, you have Tmax, and then T half. So as the graph shows, the Cmax, it is the once you take any bioactive component, what is the maximum concentration of the bioactive component achieved in the blood or your serum. So that is your Cmax. And then what is the time taken for that concentration to achieve. So that is the Tmax. And then how much time it is required for the concentration of the bioactive components to reduce from the Cmax to half of it. So that is the T half. So these parameters, they are really very important, because if you are taking a bioactive component and its Cmax, it is really high, because you know that the bioactive components, they are just like drugs, the nutraceutical, they are just like drugs. So at lower concentration they have one effect, at higher concentration they have another effect. So if a bioactive component, it attains the Cmax very quickly, and very high Cmax value it is there, so then it might affect adversely. So that's why these things, they need to be taken into account. And similarly, the T half, it is also very important. So if Cmax it is achieved, but immediately it is removed from the system through excretion or metabolism, so that means its T half, it will be very less. So the bioactive component, it should be available for sufficient period of time, only then it will be able to provide its desired effect. So if it is not available for that duration, so the effect, it is not going to be there. So that's why the T half, it is also very important. So let's see now what this bioaccessibility, absorption, and the transformation they are, because ultimately you are going to represent the bioavailability in these terms. So bioaccessibility, as I told you, that there are three factors which are influencing bioaccessibility: liberation, solubilization, and interaction. So I'll just explain you with the help of the example, like say anthocyanin and the carotenoids, they are embedded in the matrix. So you require different kinds of processing method. So I'll give you the experience of my lab. So we were working on the anthocyanins, and initially the method which we are using for the extraction of the anthocyanin, it was having very low yield. But then we shifted to microwave assisted extraction, the ultrasonic extraction, and then finally the enzyme assisted extraction. So we used a enzyme known as the visco enzyme. Visco enzyme, actually it is not a single enzyme, it is a mixture of various hydrolytic enzymes, so that can break the cellular matrix, and that in turn will release the anthocyanins from the matrix, so thereby making it more bioaccessible. And the interactions, they are also very important, because for example, the omega-3 fatty acids, they have the ability to link with the calcium ions. So if you are taking omega-3 fatty acid in milk, which is a rich source of the calcium ion, so you are not going to get the actual benefits of the omega-3 fatty acid, because these fatty acids, they will interact with the calcium in a very stringent way, and thereby converting themselves into the salt form, and that salt form, it is not bioaccessible, it is not going to be permeable further, and that's why its bioavailability, it is going to be less. And then coming to the absorption, so you can see that in the slide it has been shown that there are these are the cells which are present uh, on the enterocytes, and here you have the presence of the mucus, which is made up of polysaccharides and the glycoproteins, and this is the bioactive component which is going to which is intending to enter inside the enterocytes. And mucus, again, it is a very complex uh, uh, environment, complex ecosystem. So if the size of the bioactive molecule, it is very high, if molecular mass is very high, so it is very difficult to pass through the mucus. And I'll discuss in the next slides how we can overcome this problem. And then in this enterocytes, the epithelial cells of the enterocytes, you can see that the transport, it can occur via paracellular means or via intracellular means. So in between these cells, epithelial cells, you have certain spaces present, and those spaces, they are known as the tight junctions. So although the size of these tight junctions, it is very small, less than 0.7 micromole molar, but not molar, microns, 0.7 microns, but still some small molecules like amino acids, they can pass through these tight junctions, and thereby making them bioavailable. So this is one way of transport. that is the paracellular transport, and then you have active and the passive transport, which is also possible. And you know what active and passive transports are. Passive doesn't require the assistance from the energy, while the active one, it requires the energy. Mm, and then another point which I would discuss about the transformation, because the transformation, as I mentioned, it could be through chemical means, or it could be with the help of the enzymes. So many a times, say this gamma tocopherol acetate, which is the form which is being used for biofortification purposes. So immediately, if it can be converted into the tocopherol, so then it is going to be more bioavailable. But transformation, it can have it is a what we say, the double-edged sword, because it can have the positive effect, it can also have the negative impacts. If so, after the biochemical modification, you have a form which is more bioavailable, so then naturally the bioaccessibility, bioavailability, it will be more. But if after the transformation you get a modified form of the metabolite, which is not able to permeate through the membranes of the enterocyte, so then the bioavailability, it is going to be less. So let's see what this bioaccessibility, I'll just explain it with the help of the examples. So as I mentioned before, the bioaccessibility is nothing but a fraction of a compound which is released from the food matrix in the gastrointestinal lumen, and thereby made available for the intestinal absorption, because this is the first step. And here for bioaccessibility, as I have written here, the liberation versus stability, because many a times if you are using the harsher conditions in order to have the more yield, this happens because many thermal procedures, the thermal processing methods, they are being used for the extraction of certain bioactive component. But those thermal processing methods, they reduce the stability, they reduce the functionality. And that's why I said before also that a balance, it has to be maintained between the yield and the functionality and the stability, of course. And then the physical form of the bioactive compound, it is also very, very important as far as the bioaccessibility it is concerned. So I'll explain it with the help of the example of the carotenoids. Carotenoids, you know that they may be present in tomato, papaya, and mango, but in all these three crops, they are present in different forms. In the papaya, they are present in the lipid soluble forms, while in the tomato, they are present in the crystalline form. In the mango, they are present in the liquid crystalline form. So this physical form of the carotenoid, it will also have influence on the bioavailability. So usually you will find that amorphous ones, they are more soluble as compared to the crystalline. The physical form of the bioactive component, it is going to have the major influence. And then choice of the processing method, as I have already explained, the thermal versus non-thermal. And then interdependence, this is again very, very important. And, a study was done on the purple and the red tomatoes, and it was found that in the purple tomatoes, the bioavailability of the lycopenes, it was much higher as compared to the red tomatoes. Because in purple tomatoes you have another bioactive molecule present, that is the anthocyanins. So these anthocyanins, they are acting like a sacrificial molecule. So before any damage it is done to the lycopene, the anthocyanin molecules, they come in the way and they get destroyed. And it is just like, say, what relationship between the vitamin C and the vitamin E. The vitamin E, we call it as a primary antioxidant, while vitamin C, we call it as the secondary antioxidant. The secondary antioxidant, that means first it will play its role, and then finally the vitamin E will come to the picture. So that, in other words, we can say that vitamin C, it is required to protect the vitamin E. And similarly here, there is a quite possibility that the anthocyanins, they are actually playing the role of sacrificial molecule to prevent the lycopene from the damage during the uh, extraction process. And then what are the factors that are limiting the bioaccessibility of the phytonutrients? So again, I am explaining it with the help of the carotenoids. If there is a restricted liberation, so then of course the bioavailability is going to be less, and solubility, the hydrophilic and hydrophobic bioactives, again both of them, they are going to have different levels of bioavailability and interactions with the co-ingested molecules. It is again very very important, because many a times in case of drugs, you might be remembering your younger days when our parents, they used to say this drug it has to be taken with the milk, this drug it has to be taken with the juice, and this one should not be taken with the milk. So all these, they have some meaning, because the component which you are co-ingesting with the bioactive molecule, it is going to influence the bioavailability of the bioactive component. So this has to be really taken into consideration. So this is how the things they happen. So bioactive component is present in the matrix, then it is released from it, and once it is released, it should be present in the stable form, and once it is stable, it should be present in the soluble form. So this soluble form, it will then diffuse, and it will be absorbed. It will enter in the enterocytes, because very easily it can pass through the membranes of the enterocyte. And the next hurdle you can see is the absorption. So as I told you that the mucus layer transport, it is very very difficult, because mucus, it is a complex ecosystem, and if size is bigger, so transport through the mucus layer, it is going to be difficult. And tight junction, as I have already explained, smaller molecules, they can pass through the tight junctions. And the passive membrane transport can occur, active membrane transport. And here I would like to mention the role of the surfactants, and very another important molecule, the piperine. The piperine, it is gaining importance nowadays in the food industry, because it is showing all kind of positive effects. It is being used as an excipient. Excipient, it is always excipient is not a bioactive compound. It doesn't have any other activity on its own, but actually it influences the bioactivity of the other components when present in the foods. So rhamnolipids, surfactants, and piperines, so they actually facilitate the transport in the passive mode. And then there are efflux transporters also. This is again very very important, because many a times what happens is, in these membranes of the enterocytes, you have the presence of the efflux transporter, which usually belong to the ABC class of transporter. ABC, you all must be knowing, the ATP binding cassette transporters. So these bioactive molecule, it enters inside the enterocyte, but these efflux transporters, they will efflux it out, so that in turn will reduce the bioavailability. So that's why many a time it is advisable to include the flux inhibitors in order to ensure the more bioavailability of the bioactive molecule. And then uh, other factors which determine the absorption, they are the chemical structure. For example, both anthocyanins and the isoflavone. The isoflavone, which is very dear to me, because I am working on this molecule since last 6 7 years. Both these secondary metabolites, they are actually present in their conjugated form. When I say they are present in the conjugated form, that means they are present in their glycosylated form. You all must be knowing that most of the secondary metabolites in the cell, they are present in the glycosylated form, because it enhances their stability, it enhances their solubility in the cell. So that's why this conjugation, it is there. But these conjugated forms, they are less bioavailable, because this conjugation does two things. First thing is, it enhances the molecular weight of the bioactive component, and second thing is, it enhances the hydrophilicity, because sugar, you know that there are a lot of hydroxy groups present, so they will enhance the hydrophilicity of the molecule. And if hydrophilicity it is more, so then it cannot pass easily through the hydrophobic uh, membranes of the enterocytes. So that's why the bioavailability, it gets reduced. So our system, it should have some mechanism to deconjugate these bioactive molecules like anthocyanins and isoflavones. Only then it will be made bioavailable. But unfortunately, in this our in our gastric tract, we don't have the enzymes which can do this deconjugation. Of course, a few enzymes are there, but their activity is very less, and their specificity is very low. And that's why this deconjugation, it does not take in the small intestine. And these bioactive molecules in their conjugated form, they go to the large intestine. In the large intestine, you have the gut microbiota present. So those gut microbiota, you all know that they are now being considered as the metabolic organs. So they are actually enhancing our functional capability and capacity and competency, because there are many biochemical reactions which cannot be carried out by the gene present in our genome, but those enzymes, they can be encoded in the microbiome. So in a way, the microbiome, it is making us complete. So is typical synergism, it is there between the our genome and the microbiome. And then the isomeric configuration of the bioactive component, it is also equally important. I'll give you the example of the lycopene. And the lycopene, if you see it in the pure form, it is always there in the all trans form. But once we consume it and then we isolate the lycopene from the serum, you will find that half of it is cis. So that means there is some mechanism in our body that is converting transform into the cis form, and then that cis form becomes bioavailable and it enters in our serum. And that means the cis form, it is more bioavailable. Similarly, for catechin and epicatechin, so these are the isomers which are more bioavailable. And equol is a molecule which is formed from the isoflavones. As I told you that in our microbiome there are certain enzymes which can convert isoflavones into the equol. The diargine reductase is there, dihydrodiargine reductase, tetrahydrodiargine reductase. A set of these three enzymes, they convert isoflavones into the equol. And equol, it is actually giving all the positive effects of the isoflavones. So it is but all human beings, they don't have the capacity to convert isoflavones into the equals depending upon their gut microbiota. So if we have in our gut the microbiota which is containing these enzymes, only then the conversion will take place. Otherwise the isoflavones, they won't be able to provide the desired health beneficial effects. And similarly, the hesperidin, you all must be knowing, it is present in the lemon and oranges. It is a kind of flavonoid, and it is available, it is more bioavailable in the S configuration. The tocopherols and the tocotrienol, I will like to emphasize a little bit more on this. Now, if you see the structure of the tocopherols, you will find that there are three chiral center present in the side chain of the tocopherols. And because of that, there are 8 different isomers of the tocopherols. But only one isomer, that is the triple R, it is bioavailable. Rest, they are not bioavailable. Uh, in the synthetic tocopherols which we are getting in the market, it is a racemic mixture. So it contains all the 8 isomers. So you see that only 12.5% of it is going to be bioavailable to us. So that means we are paying 8 times more to get the desired effect. And that's why more emphasis it should be given on the natural vitamin A, which is isolated from different oilseed crops like soybean. And tocotrienol, they are the cousins of the tocopherols. So they are the saturated form of the tocopherols. So here's the side chain, it is saturated. And because of this saturation, you have only one chiral center present. And because of this one chiral center, the number of isomers, they are two only. And that's why the bioavailability of the tocotrienol, it is much more as compared to the tocopherols. And now coming to the third part, that is the transformation. So you can see here the transformation, it could be chemical transformation or it could be biochemical transformation. And biochemical transformation, it could be phase 1 metabolism or the phase 2 metabolism. The phase metabolism is nothing but through oxidation, reduction and hydrolysis, we are preparing the molecule for the phase 2 metabolism. And in the phase 2 metabolism, we attach different different types of groups. It could be conjugation, sulfation, methylation, or glucourinylation. So that will have the impact on the bioavailability of the bioactive component. And then, sorry. So coming to the gut microbiota, as I told you that the gut microbiota these days, it is considered to be a metabolic organ in the human body. So if this stats, they will give you the idea about it, why it is now being considered as the metabolic organ. Because if you can see the number of cells in the microorganisms, they are 10 times more than the human cells. And if you see the number of genes, they are 150 times more than the one which is present in the human genome. So that's why they are enhancing the metabolic capacity of the human beings, because they are involved in the synthesis of the enzymes which human beings, they are not able to synthesize. And that's why a lot of emphasis it is being given on the gut microbiota these days. Even in this COVID-19 times, I found one very interesting publication. In that, they what they mentioned was that we are advocating the social distancing, we are discouraging the handshake, but uh, microbiota, it is getting exchanged between the individuals if they are nearby, if they are interacting with each other, if they are going for the handshakes. So if you maintain the social distancing, if you don't allow the handshakes to take place, so naturally the exchange of the microbiota through skin and through breathing it is going to be not there, it's going to be lacking, and that may have the influence on the health of the human beings. So this kind of study is also being done. And then apart from this, you have different kinds of functions being performed by the gut microbiota. Here I have highlighted the bile acids, because it is very, these are the only metabolites which are considered as the transgenomic. So two bile acids, they are synthesized by the human genome, and then they are modified by the enzymes and coded by the gut microbiota, thereby leading to the conversion of those two into the another two types of bile acids. So bile acids, or the bile salts which we are getting uh, uh, in our body, so they are actually being formed through combination of the microgenome and the human genome. So that's why they are considered as the transgenomic metabolites. So just like the Rubisco, which is a transgenomic, here the chloroplast genome as well as the nuclear genome of the plants. So these are the some of the positive effects provided by the gut microbiota. So it helps in the immunity, production of the vitamins and the metabolism. It reduces inflammation, obesity, and but again there are good quality microbiota and there are bad microbiota. So a balance it has to be maintained between the good microbiota and the bad microbiota. If balance is shifted towards the good microbes, so then it will have the positive health beneficial effect. If it is shifted towards the bad, so then it will have the negative impact. And then disbalance between the two, that situation it is known as the dysbiosis. So these are the enzymes which are being produced by the gut microbiota. So beta glucosidase, as already mentioned, that it leads to the deconjugation of the conjugated forms of the secondary metabolites like anthocyanins and the flavonoids. And beta glucuronidases, these are the enzymes which convert uronidated form into the free forms, and that also has the influence on the bioavailability. And I told you about the prebiotics because we these days lot of emphasis is being given on prebiotics, because they are actually the food for our gut microbiota. The gut microbiota loves that prebiotics. So it could be resistant starch, it could be soluble or the insoluble fiber, and they convert them into the acetate, propionate, and the butyrate. So these three short chain fatty acids, again, they have many health beneficial effects, because they can act as the signaling molecule. They can enhance the harvesting of the energy from the diet, and then they can also influence the acid, because they can have the effect on the certain enzymes like histone deacetylases. So through these actions, they are being involved in various health beneficial effects. And then what are the inhibitors of the nutrient bioavailability? You all must be knowing about the phytic acid and the oxalates, how they chelate the metal ions and thereby reducing their bioavailability. And similarly, the phytosterols, because sometimes the receptor of two molecules, it is same. The phytosterols and the cholesterol, both they are being made bioavailable through a common mechanism. So if phytosterols are there along with the cholesterol in our diet, so they will inhibit the absorption of the cholesterols, and that may have reduce the bioavailability of the cholesterol. And then the binding of nutrient in a form that is not recognized by the uptake system. So this is again we have discussed in the transformation system, like say fatty acids, they form complex with the calcium ion. And then enhancers of the nutrient bioavailability, so, the fats for carotenoids. So this is very very important. So if you really want to derive benefit out of the consumption of the carotenoids, so it is always advisable to take it along with the fats, because fats, what they do is, if you take carotenoids along with the fat, it will induce the production of the bile acids. And bile acids, then they will be involved in the formation of the micelles, and the carotenoids, they will become part of that micelles, and thereby enhancing the bioavailability of the carotenoids. And similarly, vitamin C, it is well known for enhancing the bioavailability of the iron. So here I would like to mention about iron as well, because iron can be made available from animal sources, from plant sources, but bioavailability from the animal sources, it is much more as compared to the plant sources, because from animal sources we get the heme iron, while from the plant sources we get the non heme iron. So whenever you have the heme surrounding the iron, so that is a kind of protective covering around the iron that prevents its oxidation. So the vitamin C, it helps in the bioavailability of the iron. And similarly, vitamin D, we all know how it is helpful in the absorption of the calcium. And then certain host factors, they also influence the bioavailability. For example, for vitamin B12, intrinsic factor, and nutrient status, and the physiological state in which we are. So that all host factors, they will determine the bioavailability. So this is as I was discussing in our lab, what we have done. So from certain gut probiotic microbes, we have isolated this beta glucosidase enzyme, and then diagen reductase, dihydrogen reductase, and tetrahydrodiagen reductase. So all these three enzymes, they are required for the conversion of the isoflavones into the coals. So what we intend to do is that either we can use these purified enzymes in the soya product, so that the bioavailability of the isoflavones, it will be enhanced, or directly we can prepare a cocktail of the microorganisms, and we can, because all these are, they are the probiotic microorganisms, so we can add them in the soya products, so that the bioavailability of the isoflavones will be enhanced, and they will be converted into the equol, because equol, it is actually more potent, and more bioactive, and its antioxidant power is much more as compared to the isoflavone. So naturally the health beneficial effect, it will be more. So this is about the bioavailability of the carotenoids. So as I told you, the carotenoids, they can be present in the oxygenated form or in the normal form. The oxygenated forms, they always have more bioavailability as compared to the normal lycopene, beta carotene, or the alpha carotene. And this is how the beta carotene, it can be resulting in the formation of the vitamin A. And I don't think whether it will be you will be interested in this part of or not, because the uh, you can see that carotenoids, because they are acting, having the provitamin A activity. So how much beta carotene you are getting when you are taking it in the with the oil, or with from the food, or whether you are taking the purified preparation. So all those things they determine the bioavailability. And then what are the other factors along with the things which we have discussed so far? The breakup from the food matrix, of course, it is important. And then the presence of the lipid, it also influences. and then lipid soluble compounds which you are consuming along with the carotenoids, so that will also have its own influence. And it has been found that apart from all these things, the dietary fiber, if you are taking more along with the carotenoids, because dietary fibers, they have ability to bind the carotenoids. So if they bind the carotenoids, so then carotenoids, they won't be bioavailable, and that's why their health beneficial effects, they will be reduced. And some, because carotenoids contain a lot of unsaturation, so if temperatures, they are too high, so that may lead to the destability of the carotenoids. And as I told you that along with the fatty acids and the bile acids, they will be incorporated into the micelles, and these micelles, then they will pass through and they will enter in the enterocytes, and finally in the lymph you will get these chylomicrons. And here, even the type of fatty acid which you are consuming along with the carotenoids, it is also very very important. So if you are taking a long chain fatty acid, the effect is different rather than if you are taking the medium chain fatty acid. Even the unsaturation, it also have a different influence as far as the micellization of the lipophilic compounds it is concerned. And this is about the bioavailability of the anthocyanins. Anthocyanins, as I told you, they are always present in the conjugated form. So these are the sugars which can be associated with anthocyanins, and depending upon the type of the sugar, the bioavailability, it will be different. For example, if you have a ramenose moiety attached with the anthocyanin, so then its bioavailability, it gets reduced, because we don't have the enzymes to remove the ramenose moiety from the anthocyanins. So that's why its degradation, it will take place in the large intestine through gut microbiota. So this is again a nutshell how actually the metabolism of the anthocyanin, it takes place. And here they also undergo in the first pass metabolism in the liver also. This is how they are different from the carotenoids, because carotenoids, they don't enter in the liver, but anthocyanins, they undergo the first pass metabolism in the liver, and that further influence the bioactivity. So, what are the strategies for improving the bioavailability of the bioactives? The first one is the chemical modification of the bioactives. I'll give the example of the curistine versus rutin. Rutin, it is very similar to the curistine. It rather, there's nothing but a disaccharide made up of the glucose and the ramenose. It is attached with the curistine, so that becomes the rutin. But as I said that we don't have the enzymes to remove the ramenose from the flavonoids or the phenolics, so that's why the bioavailability of the rutin, it is less as compared to the quercetin. And similarly, as I told you, sometimes the bioavailability, it is compromised through efflux transporters. So if we can use the inhibitors of the efflux transporters, so then bioavailability, it can be enhanced. And encapsulation, it is very very important, because uh, till the time the bioactive molecule reaches its physiological site of action, it should be stable, and unless until you encapsulate them, the stability, it will be compromised. So that's why the encapsulation, it is very very important. And then you can design various colloidal system. You can make use of the nanotechnology, because this is another area which we are venturing into now. Because the nanotechnology, I'll explain in the first slide, in the next slide rather, the neosomes. Now we are working on that, because one project we have proposed recently, in which we are intend to do the biofortification of the curd with the beta carotene and the iron. Because iron, you know, it is hydrophilic, beta carotene, it is hydrophobic. So earlier it was difficult to do the biofortification of both of them in the same product. But now with the use of neosomes, neosomes are nothing but they are made from the surfactant molecules. So by making use of these lipid and surfactant based polymers, you can develop different kinds of delivery vehicles for these bioactive molecules, which will not only ensure the stability, but they will ensure the functionality of the bioactive components as well. And some of them, especially the biopolymer based, the polyelectride complex, here what we are doing is we are simply mixing a biomolecule which is having the negative charge with the biomolecule which is having the the positive charge, say protein with the carbohydrates, and you are leading to the development of the polyelectrolyte complexes. So they are biodegradable. So this is one thing positive about these kinds of vehicles, because then the residues uh, uh, will not be a problem after the bioactive molecule it has been released. So these are some of the desirable characteristics which should be there in the delivery system, because delivery system, it should be food grade, because this is very very important. It should be in the grass category, and bioavailability, it should be more, holding capacity should be there, and controlled release should be there. So this is again very very important, because sometimes what happens, there is a burst release. That means all of a sudden a very high concentration of the bioactive component it is released, but that is not desirable, because as I told you, that effect of bioactive components, it is concentration dependent. So if very high concentration is achieved, so that may negatively impact the system. So then the use of mucolytics. So this is again recently the new area which is being taken up. That mucolytics, as I told you, that mucus, it is a very complex ecosystem. So many molecules, they are not able to cross this hurdle. So if you can have certain enhancer, the mucolytic agents like say bromelain, papain, they are very common proteolytic enzymes from the pineapple and papaya. uh, if you include those, so they can degrade, they can loosen up the mucus, and thereby ensuring the bioavailability of the bioactive components, the safe passage through the mucus, it will be there. And then there are certain permeation enhancers like say sodium caprate, chitosan, sucrose esters. If you include in your formulation these permeation enhancers, so naturally the bioactive components, they will be more bioavailable. So with this, I come to my last slide, and I would like to conclude that. So by this time, I think you all must have realized that the functional foods or the nutraceuticals, they offer a very great opportunity to prevent onset and escalation of the lifestyle associated diseases. And then the innovative oral delivery, because this is very very important, because you may put bioactive components in your diet, but unless until it reaches the site of action, it is not going to be beneficial. So that's why the delivery vehicles, they are very very important. And these days lot of emphasis is being given on mucolytic and permeability enhancer, but in near future we have to study the safety of these. Although the people, they claim that they belong to the grass category, but still we have to ensure that they don't have the negative impact on the health of the human beings. And then understanding the metabolism of the bioactives, this is also very very important, because as you know that once they are metabolized, so their functionality, it gets altogether changed. So sometimes it gets enhanced, sometimes it gets reduced. So unless until we have the information available as regard to the metabolism of the bioactives, so it is very difficult to predict their bioavailability. So unless until we have the clear picture about the bioavailability of the bioactives, you cannot go for the recommendation. So just like other nutrients, we have to give the recommendations about their dietary, what are the RDAs, or recommended dietary allowance, what it is. So that will be possible only if we know about their metabolism, and that will ensure that we get a clear picture about the bioavailability of these bioactives. And then in vitro and in vivo models, they need to be developed, because in the one of the slide I mentioned that the uh, uh, we are not able to win the trust and the faith of the customers, because there is not sufficient scientific data to fortify our claims. So that's why advanced procedures, they are required. We need organs on chip. And C. elegans, it is now emerging out to be a very good system for and studying the bioavailability of the bioactive components. So with this, I thank you all for the patient listening.
Thank you, sir. Thanks for your nice presentation. There are many questions in the chat box, and most of the chat box is filled with the nice presentation, like comments. Thanks for that. First of all, I will ask one question from Dr. Vikas Pawar.
Yeah.
Uh, which is anti-nutritional factor of pearl millet and how to improve availability of grain micronutrients?
Because in pearl millet, you know that C glycosyl flavones are there, and then there is concentration of the tannins also present, and even lipoxygenase level it is very high, so that is responsible for the reduced shelf life of the pearl millet flour. So although we don't consider them as the anti-nutrition, but of course the C glycosyl flavones they are being considered as the even phytic acid concentration it is also very high in case of pearl millet. So these two, the phytic acid and the tannins and the C glycosyl flavones. So these are the three anti-nutritional factors which are present in the pearl millet, which need to be reduced if we really want to enhance the consumer acceptability of pearl millet.
Thank you, sir. Dhananjaya Kolar, which management practice increases the bioavailability of nutrients under water stress conditions?
You're talking about the plant nutrients.
Yes, sir.
Because and in the water logging conditions, of course the oxygen availability it is going to be less, and the role of the soil microbiota it is going to be minimal, so that those things that we should be taken into account while devising strategies for enhancing the availability of the nutrients from the soil.
Uh, what are uh, what are the sources of phytonutrients apart from plants?
No, no, as the name suggests that phytonutrients means
Yes,
they come from the plants.
plants only.
Mm. No, but of course, these carotenoids, even in the algae, you will find the carotenoids.
Yes, sir.
Even in the microorganisms also, you will have all these things. Even rather, now a group it is working on a heterologous expression of the genes, thereby incorporating the whole pathway for the synthesis of these phytonutrients in the microorganisms, whether it is the fungi or the bacteria. But that is through genetic means, but naturally also in certain algae you have the presence of these nutrients apart from the plants.
Okay, sir. Shresha is asking about what are the bioactive nutraceuticals reported to prevent carcinogens. Please list, if any.
Now, as I said, that in the literature, flood of information it is there. So you name a disease, you name a bioactive molecules, you will find the correlation between them. One or two papers they will be there. So all these, whether the carotenoids, the anthocyanins, or the phenolics or the flavonoids, so all these they have been linked with the carcinogenic properties. Of course, the glucosinolates, one which are present in the brassica, they have also been linked with the anti-carcinogenic properties.
Okay,
Apart
sir.
from these,
Thank you, sir. Rest of the questions we will answer through mail. And now I would like to request Dr. Lokhande sir to propose vote of thanks for this program.
Before that, I would like to say something. Can I?
Yes,
yeah,
sir. Yes, sir.
Dr. Dahuja, kindly accept my compliments for a very nice presentation,
thank you
which
so much.
covers because this is the need of the hour.
Yeah,
Everywhere we see that biofortification programs are going on, and people think that if you produce a nutrient dense crop, maybe high Fe, high Zn,
mm,
and that will solve the problem. But bioavailability is a real concern,
yeah,
and we are more interested so far as the pearl millet is concerned, because here a lot of people in Rajasthan and Maharashtra, they consume bajra as a
Yeah.
staple food, you can say, and the iron-rich bajra, iron-fortified bajra is variety this university has released, which is having around 80 to 85 ppm of iron. So, can we determine the bioavailability of the iron of the variety? That
Yeah,
is my
yeah.
question.
And that's
Of the
why
floor, you can
that's what I
okay.
discussed in my presentation also, that now
Uh-huh.
you have the in vitro simulation models available. So
C.
through in
elegans.
vitro simulation models, you can like say Infogest is one model, but all these
Okay.
model, they are the static models. So a
Okay.
static model means because when we are consuming any food, so in our gut there is a peristatic moment going on.
Okay.
You understand the peristaltic movements of the organs and the it is there.
Okay. Right.
So
Hmm.
in the in vitro conditions, what we are doing is we are using the static model. In the static
Okay.
models only, we are giving the enzymes which are produced in our body, but the movement which is there in the body, that is not being simulated. So
Correct.
those models, they are known as the dynamic models. So till now we have the static models of in vitro models available, simulation models, so which can be used for determining the bioavailability of the iron. But that will give you a rough idea. But if we really want to go for uh, uh, the real determination of the bioavailability, so as I said, the organs on chips, it is one new field which is coming out, and then the dynamic models, they are being developed. So they, where they are actually recreating the environment which is there in our body, including all those movements, the peristatic
Okay.
movements. And then C. elegans, because it is having the same kind of organs uh, as the human beings, so at least in
Hmm.
part. So C. elegans, it is being considered as the another option for determining the bioavailability. But at present, we have only the static in
Okay.
vitro models,
Okay.
simulation models to for
Because
determining them.
okay. Okay.
Anything else,
Uh,
sir?
Thank you, sir.
yeah, Dr. Kaushal, I just would like to clarify one doubt
Yeah.
uh, regarding dietary supplements
Mhm.
or intake of purified bioactive compounds. What is your opinion regarding the intake of dietary supplements or maybe the bioactive compounds in the purified form? That is the only.
Uh, and as I said that because when a bioactive component, it is part of the food matrix, so it
Hmm.
can have both type of effect. It can have a positive influence, it can have a negative influence, because the molecules which are co-ingested in the food
Mhm.
matrix, so they may act as enhancers or they may act as the inhibitor. So, so if you have a bioactive molecule which is present in a food matrix where it is co-ingested with the enhancers, so
That
naturally,
is
when you are taking the supplement, the bioavailability of the supplement will be less. But if you are
Mhm.
taking a bioactive component which is co-ingested with the with an inhibitor, so in that
Mhm.
case, if you are taking that bioactive component from as the supplement, the bioavailability of the supplement, it will be more as compared to the natural one.
Okay. That depends. Okay.
Yeah, yeah.
Thank you. Okay, sir. Thank you. Thank you very much and excellent presentation. Thank you.
Thank you, sir.
Huh. Yes, Prashant.
Thank you, sir. Dr. Lokhande, sir, please.
Very good afternoon to everyone. Respected Dr. R.M. Naik, sir, Organizing Secretary, Head Department of Biochemistry, MPK Veravuri. Dr. S.G. Gorantivar, Co-Convener, Principal Investigator, Cast Kasam, MPK Veravuri. Dr. P.L. Kulwal, Joint Organizing Secretary, and all participants from national and international organizations. It gives me an immense pleasure to propose a vote of thanks on this occasion. On the behalf of this university and our organizing committee, I extend deep sense of gratitude to Dr. Anil Dahuja, sir, Principal Secretary, Principal Scientist, Division of Biochemistry, IRI New Delhi, for accepting our invitation and giving very much informative and in-depth talk on even at xenobiotics, probiotics, prebiotics, and nutrient absorption, and during this COVID-19 pandemic situation. Thank you very much, sir. I extend my sincere thanks to Dr. Prashant, Dr. Nandalal, Dr. Mohsin for their kind support. I thank all participants and faculty from national and international organizations who are actively participated in this webinar. With the permission of organizing secretary and co-convener, shall we close today's second session?
Yes, sir. Yes, sir. Thank you.
Thank you. Thank you, sir.
Chan.